Hello, everybody, and welcome to this installment of the Cosmos ID webinar series, today featuring the oral microbiome. My name is Arne Materna. I'm Vice President of Product at Cosmos ID, and I'd like to thank you all for dialing into today's webinar. Please use the chat function to submit us your questions, and also take a look at the section in which you can find handouts such as literature and other references. It is my distinct pleasure to introduce to you today's presenters, both very distinguished researchers who have made impactful contributions to our understanding of the oral microbiome. Professor Deirdre Devine is Chair of Oral Microbiology at the School of Dentistry, University of Leeds, and Professor Nizar Al-Hebshi is Associate Professor and Director of the Oral Microbiome Laboratory at the Temple University School of Dentistry. A heartfelt thank you to both of you for joining us today. The oral microbiome is one of the most diverse and unique microbial communities in the human body. In fact, the Cosmos ID R&D team makes significant efforts to optimize both sequencing and analytical methods for different specimen types and body sites. And as you can see here on the right-hand side, a chart that shows microbiomes from different body sites based on their alpha diversity on the x-axis, so alpha diversity or species richness, and on the y-axis, the expected background of human DNA. And it becomes apparent that the oral cavity is divided into a range of unique habitats, such as saliva, the tongue, the buccal mucosa, teeth surfaces, gums, the palate, and so on, all of which can be sampled individually or jointly using, for instance, oral rinse techniques. Another distinct feature of the oral microbiome is its very high alpha diversity compared to other sample types. Here are a few stats on the oral microbiome. 1 milliliter of saliva contains, for instance, 100 million cells. And today, about 700 distinct prokaryotic taxa have been described in the context of the oral microbiome. However, a healthy oral cavity typically only harbors 100 to 200 taxa. While the oral microbiome is quite understudied, for instance, compared to the gut microbiome, as you will hear in today's webinar, the oral microbiome has been associated with a range of diseases, not only including oral diseases, but also systemic diseases such as colorectal cancer, pancreatic cancer, cystic fibrosis, cardiovascular diseases, rheumatoid arthritis, Alzheimer's disease, diabetes, and several others. It is, in fact, this association with health and disease why Cosmos ID has engaged actively in oral microbiome research. This slide just features a few of our recent contributions to this field. A full list of publications that we had the honor participating to can be found under www.cosmosid.com/literature. Cosmos ID attempts to contribute to microbiome research end-to-end. While our next-generation sequencing and service laboratory, which are CLIA and GLP grade, offer next-generation sequencing services, we also offer bioinformatic analysis services and software products, all focused on microbiome analysis. Since microbiome analysis is uniquely vulnerable to the introduction of bias or contamination, we use a highly controlled and standardized process throughout sample collection, storage, processing, sequencing, and analysis. And we have also adopted a range of different laboratory standards and regulatory standards. But what Cosmos ID is probably best known for is our industry-leading platform for strain-level microbiome analysis. We have curated what we believe is today the world's largest and most comprehensive database of continuously curated and phylogenetically organized biomarkers. It is, in fact, this phylogenetic organization of the genomic biomarkers that enables the accurate strain level resolution that the Cosmos ID platform is known for. Cosmos ID can not only detect bacteria, but really all kingdoms of the microbial tree of life, including viruses, fungi, protists, as well as a range of different characteristics such as antibiotic resistance and virulence. To ensure continuous performance and high specificity and sensitivity, we engage in in-house benchmarking using published best practices, but we also submit the platform continuously to blinded, independently carried out third-party validations, all of which have attested the Cosmos ID platform industry-leading performance when it comes to strain-level microbiome analysis. With that, I'd like to give you a very short and also incomplete preview of the Cosmos ID platform. Clients who sequence with us are familiar with the platform, as the results are delivered through this platform to them. However, it is as a standalone platform, really available and accessible to everyone. You can navigate to app.cosmosid.com, create your own account, or sign into your existing account. Once signed in, you can organize and dive deeper into individual samples. In this case, we're selecting a shotgun metagenomic sample from the healthy oral cavity, which is then presented in various formats. This tabular format shows you strain level resolution with the individual strains at the left side, together with their tax IDs, values for absolute and relative abundance, and a range of other statistics. The table can be filtered to exclude potential false positives, and it can be aggregated to higher taxonomic levels. The visualizations can also be switched to widely used interactive and intuitive visualizations used in microbiome research. And it is furthermore possible to set up comprehensive multi-cohort analysis, comparative analyses. Here we're looking at a data set published by one of our speakers, focused on the development of caries. As we're cycling through different tabular and visual outputs, such as heat maps, stack bars, beta diversity, principal coordinate analysis, we arrive at alpha diversity, which clearly shows that the development of caries is associated with the colonization of the oral cavity with an increasing number of taxa. However, with this, I shall conclude this short introduction and demo on Cosmos ID and hand over the stage to our first presenter. Deirdre, the stage is yours.
Thank you very much, Arne. And thank you to Mo and Cosmos ID for inviting Nizar and I to present today. So I'm just trying to get this up to presentation. So I'm just going to give a brief introduction to the mouth as a microbial habitat and the oral microbiome. Arne's already said that the mouth is a very heavily colonized site, and there are up to 10 to the 10 bacteria in the human mouth, with one individual carrying maybe 100 or 200 species at one time, depending on health and the condition of the mouth. The mouth has the most accessible and complex biofilms of relevance to human health, and they're pretty well characterized. We can grow, or people can grow, 60 to 65% of the organisms that we know exist within the human mouth, and that's pretty good compared to many sites. With the advent of techniques such as next generation sequencing and detailed metagenomic, transcriptomics, and other omics studies, we have a pretty an increasingly in-depth understanding of resident microbial communities and the development and the finessing of paradigms that are associated with the mechanism underlying disease development. We've also got a lot of in vitro, in situ, and in silico models that are available to us to enable us to explore the development of biofilms and the mechanisms that maintain health or tip the balance into disease. And Nizar is going to be talking about one of those models after I finish. So the oral biofilms and oral communities are in a homeostatic balance with the host in health, and they're important to host. They're important to maintaining the health of the mouth, as well as potentially other sites. So, for example, salivary glands and organisms that colonize the back of the tongue, streptococci, actinomyces, Veillonella, are important in the nitrite, nitrous oxide cycling that goes on with the body, which is also important for cardiovascular health. But they are involved in some very important, if not life-threatening diseases. I mean, they're not life-threatening, but they are nonetheless important. So caries, according to the World Health Organization, is the most prevalent infectious disease worldwide, with very, very significant consequences in terms of well-being and cost. Periodontal diseases come sixth on that list, and about 300 million people across the world are affected by periodontal disease at one time. And we're also seeing increasingly links between periodontitis and systemic diseases such as cardiovascular disease, diabetes, rheumatoid arthritis, all of which Arne mentioned a little bit earlier, and which I'm going to be talking about again a little bit later. So the mouth presents a wide range of environments for microorganisms to colonize. So it's warm, moist, it's nutritious, it's welcoming to very extensive biofilm formation across the various sites that you find within the mouth. Those organisms are subjected to innate and adaptive host responses with which they've co-evolved and with which, again, in normal circumstances, they're in a homeostatic balance. And you get a rich array of organisms. So bacteria, eubacteria are the most numerous, but you also get viruses and particularly bacteriophages, which may well outnumber the bacteria, fungi, in particular Candida, for example, and you can see protozoa grazing on these microbial communities within the mouth. So the sites for colonization include mucosal sites, some of which are keratinized, so like the gingivae, the gums, and the hard palate, and then the non-keratinized surfaces like the cheeks, the lips, and the soft palate. But then the mouth is really unusual in terms of the human body in having, in microbial terms, huge permanent non-shedding surfaces, which are the teeth. And these abut onto mucosal surfaces at these gingival margins. So that's very unusual, and that provides an increased array of habitats that microorganisms can colonize. So if you look at the mucosal surfaces, in sort of this domain within the mouth, saliva is a major influence on the communities that develop. So salivary flow rate will impact on those populations, and there's a lot of variation between individuals in terms of the speed of that flow rate. Saliva contains mucins, which will carry away organisms that have been aggregated within the mucin. It contains defense components like lysozyme, yeah, so that's a major shaper of the populations that are in that supragingival and the mucosal domain. The microbiota associated with a lot of mucosal surfaces are relatively low diversity and dominated by streptococci, but you can get some of the anaerobes that we associate with periodontal disease existing and persisting by becoming intracellular within epithelial cells, sometimes piggybacking on the back of other organisms to which they adhere. The tongue is another quite complex environment for microorganisms. It's highly papillated, so it has lots of these papillae, and again, in microbial terms, these create massive sort of structures that organisms can colonize down to the bottom, so they can become very anaerobic. And you can get a more diverse microbiota associated with the tongue, more diverse and more numerous. So those streptococci and the gram-positive Actinomyces are joined by other organisms that include some of the gram-negative anaerobes that you see subgingivally. With the teeth, as I said, it's unusual in having this permanent non-shedding surface, but what that creates is another variety of sites that organisms can colonize, and these can include sites that can become quite stagnant. So you can get quite different populations gathering in these stagnant sites, and you can get very diverse microbiota associated with teeth, but with quite a lot of site variation as you go around the teeth. So you have you have sites that are supragingival on the tooth surface where supragingival plaque can accumulate. Plaque can accumulate in relatively protected sites between teeth, so forming a proximal plaque, and within fissures, in fissure plaque, where again you can get some quite protected and potentially stagnant sites. And then where the tooth meets the gum margin, you get the collection of subgingival plaque, which forms as supragingival plaque extends down the tooth towards that gum tissue. And this can develop into what are called periodontal pockets if you get inflammation and disease, and these can be as much as 12 millimeters deep, going down the tooth root. And these paper points will extend quite far down within that periodontal pocket. So that's a very stagnant site which can become extremely anaerobic. In the supragingival domain, saliva is a major influencer of these populations, but once you get into the subgingival domain, then the populations are influenced by gingival crevicular fluid, which is a plasma-like or a serum-like exudate, which slowly flows into that gingival crevice, and that flow increases as you get inflammation and tissue damage when there is disease. So we know that oral sites are colonized by complex communities, and that complexity and the diversity depends on what site we're talking about, and the populations are site-specific. The Human Microbiome Project included sampling of 9 oral sites, so there's a lot of data about the relationship between the populations at each of these sites and how related they are to each other, or how different they are to each other. So they are site-specific, but there is some interdependence. So, for example, I've already said that supragingival plaque develops into subgingival plaque. The saliva, the population within saliva is collected from other surfaces within the mouth, so that salivary populations will reflect those other surfaces. The tongue is seen as a reservoir for re-inoculation of organisms into supragingival and subgingival plaque. So whilst they're separate and they're shaped separately, there is some interdependence and interactivity. So when the mouth is colonized, it's colonized by communities. Like I say, they're complex, but the interactions that underlie the development of these communities is relatively well characterized. So the communities form through sequential and specific interactions, and this is a classic figure by Paul Collenbrander, who he and his group had a series of studies where they came up with this. I'm trying to find the word to describe it, but this is something that you'll see in textbooks and a lot of us use. So what happens, for example, on the tooth is that the tooth surface acquires a pellicle, which is derived from host substances, so salivary or other molecules will attach to the tooth surface, and it's this pellicle that oral organisms then adhere to. So the primary or the pioneer species are predominantly streptococci and some actinomyces, and these will stick to the acquired pellicle. And again, these are specific interactions. Once they've adhered, they then present a different surface or a new surface to another round of incoming organisms, some of which can attach to these primary colonizers. Once they grow, or once they colonize and they start growing, they will change the environment. So they change not just the surface, but they also change the environment and the biochemical balance of the environment. So colonizers that come in and stick to them will also metabolize. So as this happens, the conditions change, which may be advantageous or disadvantageous to subsequent organisms that colonize or try to colonize. And you have organisms like Fusobacterium nucleatum, which is a really important organism in terms of maturation of plaque. And it's a promiscuous adherer to a wide range of other organisms. So that forms coaggregation bridges between some of the earlier colonizers and the later colonizers. So what you have is this sequential and well-characterized specific interactions that result in the buildup of plaque. And once that starts, you get a series of successions. So each set of organisms change the conditions, allowing succession to another population, until eventually you have a more diverse community, which has the gram-positive rods and cocci, but also gram-negatives and anaerobes. The populations and the way they succeed each other, this is shaped by the source of the colonizing organisms, by the interactions of the biofilms with the host, and also microbe interactions within those communities. And you eventually get the formation of a climax community. So this is a mature community, which again is ordered. Its structure is dictated by those sequential interactions and the way that those organisms then interact with each other. And it has a 3D structure. And this bottom figure on the right is one that we took of plaque that was formed on an in situ enamel device that we stuck onto the tooth of very amazingly willing volunteers. So this is formed in situ in the human mouth, and you can see there's a 3-dimensional structure, and you get channels and pores through the mature plaque. So whilst it's a climax community, it's not a static, it's a dynamic community. So it continues to respond to those changes within the organisms that are in it. There's clonal turnover, and it will be responding to external stimuli and host defenses. But it is pretty stable, and anybody who's worked with probiotics or who's read about probiotics will know that it's very, very difficult for an external or an extraneous organism to establish itself permanently within a stable climax community within the mouth and at other sites within the body. We said earlier that there are a large number of species within the human mouth, but it's important, I think, to sort of say that this is sort of dominated by a relatively smaller number of genera and species. So when you start looking at the profiles, and this is a slide that Nazar kindly let me use, you have a long tail of organisms that are in lower abundance, and the higher abundance, there's a set of species and genera that are more dominant. So in terms of the way that these populations develop over time, we acquire our oral microbiome very early in life, so during birth, and there are documented differences between the organisms or communities that colonize babies, depending on whether they've had a vaginal birth or Caesar-section birth. So you acquire organisms, or babies acquire their organisms from the mouth from a variety of sites, and also from close carers and siblings, from breast milk and also other food sources. So you get organisms that colonize mucosal surfaces. So again, a lot of streptococci, but that will change once you get the eruption of teeth. So you get the generation of those other sites that the teeth provide, and you get greater diversity and colonization by organisms that inhabit that variety of sites. So the establishment of that stable ecosystem evolves during a period where there's relative plasticity and variation, and mainly during the first 2 years of life. And that there is plasticity as children develop, but by probably adolescence, it's really pretty stable. Over the years, that microbiome will develop, and its composition will be influenced by genetic variation and immune status. Socioeconomic status is a very strong influence on the development of the microbiome and development of disease or not. Hormones will affect it, smoking, drugs for leisure or medicine, and the diet and the kind of diet that people have. And then eventually other significant changes happen. As we get older, our host responses change, and there's a lot of evidence of a decline of host responses in old age, and the colonization by organisms that you don't normally expect to find in a healthy mouth of somebody who is younger. When you lose teeth, you lose those organisms that are dependent on teeth for colonizing. But then you can also change the environment again by introducing other surfaces like dentures. And don't tell her, but this is my mum, somewhere around her 90th birthday, about 4 years ago. But she's of the generation where for her 19th birthday, she was given a present of having all of her teeth removed to preserve her health in later years, and also to give her a nice set of bright dentures to smile at people with. I think it's true to say that we know that the populations associated with babies, children, early years of life, and those in later years are pretty well studied. But I think middle years are less well studied. And I think there is a question that some people are now asking about the development of those, or the behavior of those populations, and whether there are ways that we can actually increase the resilience of the oral microbiome in these years to protect against those diseases that you associate with older age. So I've been talking mostly there about what's normal and what you find in health, but you get, as I said at the beginning, important diseases caused by oral biofilms, and these generally result from ecological shifts. So, and a breakdown in that host microbe homeostatic balance. So in caries, the ingestion of sugar in the diet that's used by groups of organisms to produce acid, which selects acidogenic and aciduric populations that cause dissolution of enamel and development of caries or holes in the enamel. The role of inflammation subgingivally to promote the development of pathogenic populations that are associated with a very proteolytic anaerobic population that cause periodontal disease, with the damage being caused largely by the host. This is a smoker and somebody who's probably got underlying other health and socioeconomic factors. So that's pretty disgusting. I should really take that out, I think. And then things like candidosis. If you upset the ecology by taking antibiotics, that can allow candida to emerge and to cause significant problems. Then there are shifts due to the introduction of new materials like dentures, which I've already mentioned, or implants and the development of implantitis. And then finally, the transmission of oral organisms to places that they shouldn't be. So through injury, for example, into tissues of the head where they can form abscesses. This is a bite wound, which is they can cause human bite wounds can cause really unpleasant infections. The most common of which are on hands, from people punching other people in the mouth. So you could perhaps say they deserve the nasty infection. And then transmission in the clinic. So both patients and dentists are subject to the transmission of oral organisms through treatment. So as I said earlier, we understand oral communities really comparatively well. We don't understand everything about them, but with the application of multiple technologies, from some really high quality culture studies that were done in the sort of 70s and 80s, and groups like the Moors come to mind. But there was a lot of really very good culture work, and this is now being expanded through the application of the omics. And this has led to a much greater in-depth understanding of microbial communities that are associated with health and disease. So for example, the communities that are associated with periodontal health, that are in symbiosis with the host in this homeostatic balance, and in something that Nazar refers to as normobiosis. Once you have an accumulation of plaque and the development of inflammation and gingivitis, you're selecting a population there that is more diverse, that's more anaerobic, that's dependent on the proteins and the proteolytic activity that they can display within the increased flow of gingival clavicular fluid. But this inflammation and this damage can continue, and in some individuals can develop into periodontal disease, where there are populations that are selected that cause extensive inflammation, that depend on the inflammation for their growth and survival. So they can be inflammophilic, and they promote they cause a deregulated inflammatory response. And there's a bit of a vicious cycle of activity going on there that produces the populations that we see in periodontal disease. And this has really allowed the application of new techniques, have allowed the evolution of new mechanistic paradigms of the way that disease develops. So for example, from the ecological plaque hypothesis that Phil Marsh coined in the 1990s, really to describe caries and then extended to periodontal disease, to the more recent polymicrobial synergy and dysbiosis model, in which Porphyromonas gingivalis in particular is termed a keystone pathogen. And this organism has a disproportionate influence on the pathogenicity of the community, and raises the hypothesis is it raises the pathogenicity of the entire community. So you get a new set of gene expression within this disease-associated population. And again, we can look harder at links with systemic disease, and I'm going to talk about that again a bit later. So thank you very much for listening, and I will hand over now to Nazar.
Thank you, Deborah. Thank you for the introduction. Hello, everyone. I'll get directly to my presentation, but I'm going to find it. I'm not sure if I'm able to share. Do you see the screen? I have a problem with sharing the screen.
Yes, Nazar, we know. It appeared for a second and is now gone again.
Let me try again, because I don't see the PowerPoint as one of the options for sharing the screen. Although let me try again. Okay, here we go.
Perfect, we can see your slides.
Do you see it now?
Yes, and now in full presentation.
Let me start with thanking Cosmos ID for inviting me to participate on this webinar. We have been collaborating recently, and we published together and I'm looking forward to more collaboration. And it's also a pleasure to team up again with Dr. Divine. We work together on the work that I'm going to present today. So I like to start with this slide because my work revolves around this. Maybe everyone working in the field of oral microbiome sort of work on the same concept, because it's increasingly recognized that dental diseases like dental caries and periodontitis are as a result of a shift from a balanced microbial community state that we refer to as normobiosis to an imbalanced community, which we refer to as dysbiosis, and this is associated with disease. And in terms of research, this is important because we need to characterize, using all these omics techniques, we need to characterize the shifts or microbial shifts associated with transition from health to disease, but also to find ways to modulate or to attempt to restore a dysbiotic microbial community to a normobiotic or to a balanced microbial community that is associated with health. And I'm going to talk about this in my part of the presentation. So let me start with a bit of introduction for those who do not know what periodontitis is. It's a chronic long-term infection of the periodontium characterized by inflammation and tooth attachment loss. And this is manifested clinically as inflammation, bleeding, pocketing, and mobility, and can ultimately lead to tooth loss. And just to recall from what Dr. Divine mentioned in her part of the presentation, this is a result of a dysbiotic or imbalanced microbial community, specifically the part of the biofilm that is below the gum margin here. So we refer to this as subgingival plaque. So the primary etiological agent is an imbalanced microbial community below the gum. So although classically periodontitis has been controlled or treated based or using mechanical debridement, so mechanical removal of dental biofilm, with this emerging concepts of dysbiosis, normobiosis, an emerging strategy for controlling periodontitis is to reverse microbial dysbiosis or reestablish normobiosis by using microbiome modulators such as prebiotics and probiotics. And many of you know that prebiotics and probiotics have been extensively investigated in the context of gut health, but their applicability to periodontal health and oral health in general has been minimally explored. And there have been many reasons for this, but one major obstacle has been the lack of a reliable in vitro tool for screening banks of potential modulators to identify those with promising activities. There are many models of dental biofilms described in the literature, starting with those that are only based on one species or monospecies biofilms, and to those that include a handful of species. For example, the Zurich biofilm, in its two versions, the one that is meant for to model the supragingival plaque with 6 species, and the other version that has 10 species, which is meant to reproduce the subgingival microbiome. And it's basically in which biofilms are grown on hydroxyapatite discs submerged in the right medium. Scientists also tried to grow more complex biofilms using clinical inocula like saliva or subgingival plaque in devices like constant depth fermenters or CDC biofilm reactors. I show one of these in this figure. The problem with these devices is that they are expensive and low throughput, and they require big amount of medium to be used. Recently, researchers sort of combined the two models in that they used clinical inocula, but instead of using these complicated devices, they again went back to the simple devices like microtiter plates and Calgary biofilm device. Now it's called MBEC. So that although these devices do not simulate the dynamic conditions of the oral cavity, the resultant microbiomes were really diverse and were close to the original inocula. And here I'm showing one of these devices, which is the Calgary device, in which the biofilms are allowed to grow on these pegs that are, in the case of oral microbiome research, that are coated with hydroxyapatite, because that makes them similar to a tooth surface. So the gap we identified in the literature was that there is no model that has been specifically developed or optimized to screen large numbers of candidate modulators, and more importantly, quantitatively assess their effects on subgingival microbiome. So we propose to develop an innovative in vitro model that involves growing dysbiotic and normobiotic subgingival microbiomes in parallel. So you have the balanced and imbalanced microbiomes grown together. And then we calculate a novel index to measure microbial dysbiosis, and that will help us assess microbiome modulators in high throughput format. And we received a grant from NIDCR to do that. And to address this goal, we divided the work into 4 stages or sub-aims. So the first was to identify the optimal medium to propagate the subgingival dental plaque, and then sort of fine-tune the growth conditions like pH, temperature, shaking, and so on, so that we can maximize the similarity between the in vitro microbiomes and the original samples. And then, because it's not very practical every time to go back to patients or to human subjects to collect samples and use them as inocula, we also wanted to test if it would be possible to reproducibly passage these microbiomes in the lab. So by storing them like on -80 and then taking them again and try to regrow them. And also we wanted to develop this tool or quantitative tool to measure dysbiosis index, because if we want to check if a prebiotic or probiotic is changing the microbiome favorably, we have to do this using an objective tool. So we thought that the dysbiotic index will be the right tool for that. So far we have completed this first part of the project in collaboration with Dr. Devine's group in Leeds, and which I'm going to share the results with you in the coming slides. And actually the work was published earlier this year in JDR. So for the experiment, we recruited 5 periodontitis patients, so patients with periodontitis or severe periodontitis actually, and 5 healthy individuals. From each of these subjects, we obtained subgingival plaque. Then we pooled them within each of these 2 groups. So we ended up with 2 clinical inocula. One represents the dysbiotic microbiome, and the other one represents the healthy microbiome. And then we use these to inoculate a Calgary device that I showed in the previous slide. And we use different types of media that I'm going to share in the coming slide, and then allow the microbiome or the biofilm to form on the surface of this hydroxyapatite coated pegs that were also treated with sterile saliva, so that mimics the dental pellicle that forms in the oral cavity. And we allowed the biofilm to grow for 7 days, and we replenished the medium every 2 days. And after the end of the period, we harvested the biofilm to check for viability. And I think this is very important, because it's not enough only to grow a biofilm. We have to ensure that it's viable, so that we can test the different agents or antimicrobials, for example. And also we check the biomass in terms of DNA yield, and then we use that DNA to do profiling using 16S ribosomal RNA sequencing. And we used 2 types of media, broadly nutrient-rich and nutrient-limited. The nutrient-rich media included DHI supplemented with mucin, vitamin K, and hemine. Modified SHI, this medium has been used comprehensively in studies or in developing oral microbiome models, and it includes these components. But we just changed a bit the formulation, so we called it modified SHI. And because based on preliminary work we did, we found that these 2 media support the growth of different groups of bacteria. So we thought maybe by blending the components from both, we would be able to maximize the diversity of our in vitro microbiomes. And because sugar is also important for the growth of some of these oral species, we test for each of these media. We tested at different levels of sugar, 0%, 0.05, and 0.1. And then we compared this with saliva supplemented with heat-deactivated human serum. And we are not the first group to try this, because basically the bacteria growing in the oral cavity, the primary source of nutrients come from saliva and from serum for bacteria in the subgingival part. And we tested different concentrations of serum, 5%, 10%, and 20%. So let's move to the results. And we start with the biomass. And as some of you would expect, we found the nutrient-rich media to result in a higher biomass in both the peritonitis and health-derived microbiomes. MSHI was an outlier. We don't really understand why, because it's again a nutrient-rich medium, but it then performs similar to the other two ones. Although adding some of its components to SBHI increased the biomass, but on its own it did not, and the amount of biomass in microbiomes developed in saliva serum were almost in the same range. Now, what is more interesting is the viability, and as I mentioned, this is very important because we have to ensure that any biofilms we develop or microbiomes we have should be viable. And as you can see here, that although these nutrient-rich media increase biomass, but when we normalize the viability signal to the total amount of DNA we had, they ended up with low viability, especially in the peritonitis-derived microbiomes, while serum or saliva serum media maximized viability, especially again in the peritonitis-derived microbiomes. And I think the reason for this is that the bacteria growing in the nutrient-rich media grow really fast, so they reach their plateau and then maybe decline phase before we even change the medium. While bacteria growing in saliva serum, they grow sort of very slow, so they don't reach the plateau before we change the medium. Now, this figure just shows the microbial profiles in the in vitro microbiomes derived from health and peritonitis and the corresponding clinical inicula. And you can see that we managed to capture most of the phyla in the original sample. Maybe with the only exception was protobacteria that was underrepresented in the microbiomes. Spirochetes did not grow well when we grew it from the health associated inicula, but in the peritonitis-derived microbiomes, at least in some of the media, particularly saliva serum, they were able to grow. And this shows the top genera that were captured in the in vitro microbiomes and the corresponding clinical inicula. This figure shows the reproducibility in the replicates of our microbiomes. So if you can see here that all the profiles were almost identical within each medium type. Now moving to what sort of differences we observed between the different media and what bacteria was enriched by which medium. So we'll see that, for example, serum enriched Performonas and Alloprovitella and Bacteroides and Treponema, while BSHI enriched Piptostreptococcus and Veillonella. Fusobacterium and Streptococcus were enriched in BHI, while Provitella were enriched or sort of overgrow in SHI medium. This shows the results at the species level, so it's more or less the same. Now let's have a look on species richness. In both the health-derived and peritonitis-derived microbiomes, saliva serum grew the most number of species compared to the iniculum. And we can see this both in the observed and the expected number of species. However, when we calculated the alpha diversity, for some reason, particularly because certain species overgrew in the saliva serum, what's going on? We can see that the alpha diversity dropped a bit for the saliva serum if we calculate the Shannon index for the healthy or health-derived microbiomes. But it still remained higher than the others for the peritonitis-derived microbiomes. Maybe the most important or interesting part of the results is this one based on beta diversity, because this sort of clusters all the microbiomes based on the overall similarity. So we can see in the first panel here, A, that what we have here in rhomboid are the clinical inicula. The one red is the peritonitis iniculum, and the one in green is the health iniculum. And you can see that all the microbiomes that were derived from this sample clustered with the corresponding sample and so on. So we were able to reproduce the differences between the peritonitis and the health-derived inicula. And when we look at the individual microbiomes grown in the different media, we can see that the closest microbiomes were the ones grown in the saliva serum. So they were very close to the original inoculum in both the health and disease-derived microbiomes. And when we measure this quantitatively in terms of similarity index compared to the inoculum, we'll see that for both the health and peritonitis-derived microbiomes, saliva serum produced the highest similarity compared to the clinical inoculum. We also calculated the dysbiosis index as the sum of relative abundances of peritonitis-associated species divided by the sum of relative abundances of health-associated species. And in this case, we defined peritonitis-associated species as all of the species that were more abundant in our peritonitis inoculum compared to the healthy clinical inoculum and vice versa. Now, applying this or calculating this for the individual microbiomes, we again see that these clustered by or clustered with the respective clinical inoculums. So if we have now the health inoculum with a dysbiosis of 0.3, and again the peritonitis-derived microbiome with a high dysbiosis index of 2.14, then the microbiomes grown in the different media also clustered close. So they also sort of replicated the level of dysbiosis in the original sample. But still, there were still differences. For example, the saliva with 5% almost resulted in a dysbiosis that is very close to the original sample. But when we grew the health-derived microbiomes in serum or in saliva serum, they sort of worsened the dysbiosis. So it's not really close to the healthy inoculum, meaning that having serum encourages the growth of some of the pathogens in the healthy inoculum, and that's why we have an increase of dysbiosis, although not to a level of the peritonitis-derived microbiome. So it's still okay. Sure. Okay. So I'm concluding. So overall, saliva serum is probably the optimal medium for modeling the subject-level microbiome by maximizing species diversity and maintaining viability while replicating normobiosis, dysbiosis. The dysbiosis index that we developed provided a valuable layer of information in addition to standard microbiome metrics and reflected more important difference between the tested media. With further optimization, we think that the model along with the dysbiosis index could be form the basis for a high throughput model for screening of microbiome modulators. With this, I would just like to acknowledge people who contributed to the work, my postdoc, my collaborators at Temple and at Leeds University and at Forsyth Institute. Thank you very much for listening. And you can take the answers toward the end of this webinar. Back to you, David.
Thank you, Nizar. So, yes, thank you, Nizar. I'm going to talk now as a representative, really, of quite a large group of people who've been collaborating together on what we call the RAP study, the Rheumatoid Arthritis Periodontitis Study. So there's a large number of people with a variety of expertise and professional roles, but it's a collaboration with the NIHR-funded Leeds Biomedical Research Centre, led by Professor Paul Emery, out of this is Chapel Allerton Hospital, and myself and colleagues within the School of Dentistry in the Division of Oral Biology, where we have our basic sciences, restorative dentistry, and our dental translational and clinical research unit, and also with colleagues in the NHS hospital, the Leeds Dental Institute, with whom we are closely aligned. So rheumatoid arthritis is a systemic autoimmune disease, and it manifests itself as a chronic polyarthritis. It affects about 1% of the population and more women than men. It's associated with the development of autoantibodies that are raised against proteins that have been post-translationally modified. So, for example, citrullination of arginine is a common post-translational modification, and anti-citrullinated protein antibodies, or ACPA. Later on, I'll refer to them as CCP antibodies. So this is anti-citrullinated protein antibodies or citrulinated peptide antibodies. These are present in about 70% of people with rheumatoid arthritis, and they can be detected up to 10 years before the onset of full symptoms. So they might have rumbling non-specific symptoms, but they don't have the full-blown rheumatoid arthritis symptoms. So there's increasing evidence that the initiating events that cause the accumulation of these autoantibodies occur at sites that are distant to the joints. So the autoantibodies are detected in serum years before joint inflammation, and that suggests that the joints are targets and not the primary sites of the cause of the disease. You get an enrichment of IgA autoantibodies in individuals who are at risk of rheumatoid arthritis. And IgA antibodies are they're antibodies that reflect mucosal immunity. So IgAs are major antibodies in secretions like saliva, bronchial secretions, including sputum, colostrum, and at the gut mucosal surface. So it's kind of suggested that a localized mucosal immune response might first develop at certain mucosal sites like the mouth, the lung, the gut, and these are important in the initiation of the accumulation of those autoantibodies that eventually result in the formation of full-blown disease. So in terms of periodontitis and rheumatoid arthritis, there are strong epidemiological evidence that periodontitis and established rheumatoid arthritis are clinically associated, and they share some common risk factors like obesity and smoking. You find, ACPA IgA have been detected in saliva, the antibodies are detected in GCF and in periodontal tissues, and these can increase in periodontitis. And there are also significant associations between ACPA seropositivity and periodontal disease. So a popular hypothesis is that in genetically susceptible individuals, citrullination associated with periodontitis might cause a localized oral mucosal immune response, which can then lead to a systemic ACPA response. And that's followed then by the synovial inflammation and the onset of rheumatoid arthritis. So this has all been studied in people who have established rheumatoid arthritis. If you look at the subgingival microbiome, there's some attractive theories that have been developed about the reason for this association. So neutrophils are an important site of the development of the antigens that result in the development of autoimmune antibodies. And the most important of these is this citrullination process, which is mediated by peptidylarginine deaminases, or PAD enzymes, of which there are 6 human enzymes. And neutrophils are enriched for PADs, and they're involved in citrullination, rather mediated by PADs, are involved in net formation and in inflammation. So this is an important process that's mediated by neutrophils, and neutrophils are also important in periodontal tissue defense. There are a couple of subgingival organisms that have been really have received an awful lot of attention because of the potential mechanistic possibilities. So P. gingivalis produces its own PAD enzyme called PPAD, and it was at one point thought to be the only bacterium that does this, but there are other Porphyromonas species that have been shown to at least express the gene. This is able to citrullate arginines that are terminal, so it works in concert with the arggingipane, which cleaves proteins at arginine sites, creating that terminal arginine, which is subject to the action of the PPAD enzyme. Aggregatibacterium actinicetam comitans, otherwise referred to as AA, produces a leukotoxin, which is also involved in the mechanistic generation of autoantigens that produce the ACPA antibodies. But I think it's important that neutrophils are very important in that periodontal environment, and a lot of organisms target neutrophils within the subgingival domain. So although these 2 organisms could have very important roles in the generation of citrullinated proteins, I think you should never forget there's a complex community at work here, and many organisms can have actions against neutrophils, so they can promote release of nets, they can affect neutrophil activity in a variety of ways. So I think you need to consider the effect of the entire community. So Prof. Emery approached us, I think, in 2015 to say that there was this link that was becoming quite strongly evidenced in terms of a link between established rheumatoid arthritis and periodontitis. But he and LBRC have a nationally recruited cohort of individuals who are at risk of developing RA. So these are the people in this area. So they might have non-specific symptoms, but they haven't yet developed rheumatoid arthritis, and a large proportion of them are autoantibody positive. So he wanted to work with us to characterize the periodontal disease status and the microbiome of those at-risk patients, really with a longer-term view to develop early interventions, to exploit this window of opportunity to see if we can slow down or stop the development from being at risk into full-blown disease. So we looked at the incidence of periodontal disease and looked at the microbiome, and were particularly interested in looking at whether there was evidence of specific roles for specific organisms like P. gingivalis. So between 2015 and 2017, we recruited to 3 groups. We had our ACPA positive or CCP positive at-risk group who had no arthritis, rheumatoid arthritis, but they have the autoantibodies. And as I say, these are from a national cohort who are recruited in Leeds and attend clinics in Leeds. We had 32 age-matched, gender-matched and smoking-matched healthy controls. And 26 individuals who had developed early rheumatoid arthritis, and they were within their first 3 months of being prescribed DMARS. So when they attended Chapel Allerton Hospital, we took over a portion of a ward and converted it into a dental space. The volunteers were asked to complete a periodontal health questionnaire. They had a full periodontal examination, which included measuring at a number of sites around the mouth the periodontal probing depth, or PPD, bleeding on probing, clinical attachment loss, and then the periodontal inflammatory surface area was calculated from the clinical attachment loss and PPD readings. So these are all indicators of the level of disease at specific sites around the teeth. 3 dentists that were blinded to the volunteers were then, they came up with a consensus decision on the periodontal status of each of the individuals in our cohort. While they were also attending, we took a number of samples. So we took saliva, serum, gingival clavicular fluid, and subgingival plaque. And we took this from both healthy and diseased sites. So up to 3 healthy sites from each person and up to 3 disease sites from the same people. And these were then analysed by next-gen sequencing in-house in the University of Leeds at our next-gen sequencing facility. And the analysis, sorry, I'll come back to the analysis in a little bit and who actually was responsible for doing that. So just a quick summary of the clinical findings. Again, just to summarise it, the at-risk group had a number of significantly higher clinical indicators and disease compared with the control group. So they had a higher percentage of sites that had relatively deep periodontal pockets, that exhibited bleeding on probing, that had a combination of a certain level of clinical attachment loss and periodontal pocket depth, which indicated that these were periodontally diseased sites. And when they also occurred with bleeding, these were active periodontal disease sites. So all of these parameters were significantly higher in the at-risk group compared with the control group. They also had a higher PISA, or periodontal inflamed surface area, and higher levels of periodontal disease. And this is just showing the consensus diagnosis of our dentists in these groups. So 73% had periodontitis compared to 38% in the healthy controls and 54% in the early rheumatoid arthritis group. There was an increased abundance of P. gingivalis at healthy sites, which was interesting, but not AA or ephilosis, which have also been associated with periodontal disease. We didn't incidentally see any aggressive periodontitis, which is the form of periodontitis that AA is particularly associated with. So we were able to do the microbiome analyses really because we not only managed to get hold of some money that would enable doing it, but we also had a very able PhD student who was supervised by Prof. Emery, myself, Josie Mead, and Tui O. Poor guy, 4 supervisors, but he managed us very well, and he's very professional. But with under Tui's guidance, he did the microbiome analyses once we'd had the sequencing done. So again, I'll have to go through this fairly rapidly. So in terms of alpha diversity, we found that at healthy sites, we are particularly interested in the healthy sites. there was significantly lower diversity in the CCP at-risk group compared to the healthy control group and compared to the ERA group. So there was a decreased microbial richness of the microbiome. In terms of beta diversity, there was also significant clustering of the microbiomes based on RA status in both healthy and disease sites. So the structure and the community structure is influenced by the RA disease status. Looking at phyla, there were, possibly not great surprises, but in terms of healthy sites, actinobacteria were the most predominant in all groups. We found synergistites were significantly higher in the at-risk group. In disease sites, the Bacteroidetes were the most abundant in the healthy control and the at-risk groups. And another significant difference here was actinobacteria were abundant in the early. Sorry, I've got that wrong, haven't I? I haven't got a star there, but anyway, I will move on. So looking at the genera that were identified, again, there were differences between the groups. So in terms of the at-risk group, there was significantly increased abundance of Bifidobacterium and Porphyromonas, which is of interest to us again, being very interested in the microbiome of the healthy sites. And in the disease sites, in the CCP plus at-risk group, there was increased abundance of Porphyromonas again, and quite a lot of differences in the ERA group. And the microbiomes associated with the early RA group do seem to be quite different in a number of ways from the other two groups. And I should say this, one thing we think this may be related to is the fact that they're on treatment, and some of the DMARDs actually exhibit some antimicrobial activity as well as moderating potentially an inflammatory response. So again, looking at the species, Jan looked at the difference in membership and structure, and you can see looking at healthy sites in the CCP plus at-risk group compared to healthy, there are quite large differences in the community membership and structure, and lower levels of differences comparing ERA to healthy controls, and then in the disease sites, lower levels comparing the at-risk group to the healthy controls. So Jan went on to look at the core microbiomes associated with each of these groups at healthy sites or at disease sites, and this was identifying species that were present in at least 80% of the samples of each group. And you can see again that it's quite an interesting Venn diagram here. So there's 81 organisms here in these core microbiomes that are in common for each of the three groups. A large number in common between healthy controls and early RA, and some that within the core microbiomes are unique to healthy or ERA, but none in the CCP at-risk group, and none that are in common with each of the other groups and the CCP at-risk group. Sorry, in common between at-risk and health control or at-risk and early RA. Similarly, in the disease sites, we get a sort of high level of commonality between the three groups, a lot of members of this core microbiomes in the early RA group, and very few that are uniquely within the CCP at-risk group. So in the periodontally healthy and disease sites, P. ginge and Arthropbacteria phenolicus, were more abundant in microbiomes of the at-risk group. So again, we're seeing differences here that stand out with the at-risk group. Jan also looked at, we sort of asked the question, are there sets of organisms that interact together that maybe can be highlighted through these microbiome analyses? And he did some network hub analyses. And I'm not expecting you to get much from these pictures, I just think they're beautiful. So I like to put them up. And what this is looking at is positive correlations between organisms and negative correlations, and also the proportion that they are of the populations in terms of abundance. So each of these dots is a node, and that represents a species and its abundance. So these were analysed to sort of get a better understanding, looking at them in terms of the top 20 nodes that were ranked, and they're displayed here for each of the groups of patients. So you can see in the healthy controls, this is the core microbiome in terms of the top 20 nodes are dominated by this group of Neisseria, and then a variety of organisms that we recognise as being sort of subgingival, so Prevotella, Porphyromonas endodentalis, Treponema vincentii, Metanarella, and a variety of Prevotella filifactor alosis. So these are all familiar subgingival organisms. The top 20 sort of network hubs in CCP plus group at healthy sites was quite different. So we have this hub of Fusobacteria, Streptococci, and then this hub here, this group here, which has some similarity to the hub that you see in the healthy controls. And in the early rheumatoid arthritis patients, it's completely different again. And disease sites, we get quite a different picture again. So the that Neisseria, also that hub that dominated in the healthy sites, has disappeared. And I think only 3 of the organisms that we saw in the larger hub have been retained in the hubs that we see here in the disease sites. It's quite interesting to me that in the CCP plus groups, that hub of 10 organisms has essentially been expanded. So this contained 9 of the 10 organisms in the CCP plus hub, and that's been extended, sort of enlarged in this hub that we see in the disease sites from the CCP plus group. And again, this contains a lot of organisms that will be very familiar to people who look at subgingival and periodontitis populations. Again, very different, and a very different set of organisms in the ERA group, but we did note that AA features as one of the dominant organisms in this hub within this ERA group, and that's really the only place at which AA has stood out for us in our studies, which is in contrast to what other groups have found when they're looking at certainly at full-blown rheumatoid arthritis. And just finally, we did do some functional analyses. So this is a metagenomic shotgun study. So this is not gene expression. This is the functional capability or potential. And there were 17 functional units that were significantly overrepresented in the CCP plus group compared with the healthy controls. When we looked, we did probe one particular functional unit, which was PAD and related enzymes, because that is a functional unit that could be looked at directly, and looked at these in the groups, the RA-related groups at healthy and disease sites. And we saw no differences in the abundance of these enzymes within those 3 groups, which is potentially surprising considering we had higher levels of Porphyromonas and higher levels of P. gingivalis. So that's quite interesting, and there's some more probing that needs to be done there in terms of not all PADs are equal. So P. gingivalis, PPAD may well be much more effective than other organisms that express a PAD. And equally, Jan Potempa's group found that there are different genotypes of PPAD, and they have different levels of activity. So I think there's quite a lot to do looking at that activity within the populations. So just to summarize quickly, we found that these at-risk individuals have increased prevalence of periodontal disease compared to healthy controls. They have dysbiotic or perturbed subgingival microbiomes, and they have an increased abundance of P. gingivalis, including at healthy sites compared to healthy individuals. So we would suggest that this dysbiosis and an increased prevalence of P. ging could play an important part in the initiation of RA, and this is something that we want to pursue further. And we particularly want to look at whether periodontitis and the associated oral dysbiosis could be attractive targets for preventative interventions, including periodontal therapy in those people who are at risk of RA, to see if we can slow that down or stop it or ameliorate the symptoms. So we're reaching out to some clinical colleagues in Glasgow and Birmingham, and we're also looking for industrial partners who might be interested in this kind of activity as well. So it just remains for me to say thank you very much to the array of people who were involved in this, but particularly to the study participants. They spend a lot of time in the chair and in clinics when they volunteer to do this for us, and we are really grateful for that. And I don't know what the time is, but I think I've probably said enough. So thank you again.
Well, thank you very much, both Professor Devine and Professor Al-Habshi, for this very, very comprehensive and informative 360 on the oral microbiome in health and disease. We've gone a bit over, but we still have plenty of attendees on this webinar. So I'd like to take a moment to thank everyone for dialing in today and for submitting a number of very interesting questions. And since many people are still on it, I think will answer a few of them. So I'll take the liberty of summarizing a couple of questions we've received from multiple people into one big one, and that is sort of the question of causation versus correlation. So many microbial taxa here have been associated, and observed in association with disease. What do we know about taxa that have been causatively associated with disease? And sort of in an extension to that question, we received many questions on how can we return a dysbiotic oral microbiome back to normal biosis, for example, through probiotics, treatments, or in nutrition and so on.
Mr. Divine?
Who wants to go first? I think that cause and effect thing is a really difficult thing when you're looking at these kind of studies, and it's very hard. Very often you see a change and you don't know if that's a result of the disease or is a cause of the disease. I think with periodontitis, there's good evidence that that the organisms actually cause the disease and they cause the disruption to the immune responses. And in terms of the studies sort of that particularly implicate P. ginge as a keystone pathogen, there's lots and lots of mechanistic stuff underlying that. So the work of people like George Hadjishengalis and Rich Lamont and Michael Curtis looking at animal models using P. ginge. The evidence, the experimental evidence for mechanisms that form part of that that theory about disease causation, that evidence is quite strong. I think with things like, we're doing an observational study with the RA where we see changes. There are potential mechanistic things underlying, but it is very difficult, and it's a very good question. And I don't know if I've really particularly answered that. I know, Nazar, I don't know if you want to say anything.
Yeah, I agree. The issue with most of our microbiome studies is that they are cross-sectional in nature, so we cannot verify the temporal relationship between cause and effect. So we don't know which came first and whether the bacteria involved or in which are the result or the cause of the result, and whether they represent. And I think probably when we look at the later stage of a disease, what we are seeing are the passenger effects rather than driver events. So maybe if we design our study to look at an earlier stage of the disease, or even before the disease starts, and we follow up, then we will be more able to look at earlier driver events rather than, and we can then verify the relationship, and we can for sure tell that this is a cause rather than effect. So I agree.
But I think that's a really important point. A lot of studies, we're looking at the end stages. You're looking at those organisms that are associated with, really damaged sites. And what we really want to know what's happening at that initiation triggering events that related to the development of perio disease. So that's a really good point, Nazar. In terms of the reversal, I think that's also a really good question. Gingivitis as a disease is reversible, so that inflammation is reversible, and that the restoring of populations is possible through oral hygiene. In periodontitis, the disease itself is irreversible, so the damage is irreversible, but you can improve things by oral hygiene and oral, sort of treatment steps that will reduce the microbial load and also take away damaged tissue. And things that resolve inflammation will also help to heal those sites. But in terms of actually restoring the population back to a normal population, there have been studies where they've looked at the recolonization of periodontal sites after they have been cleaned, and you get re-establishment of a microbiome relatively quickly. And I think.
And this emerging concepts were the emerging concept of using prebiotics and probiotics and probably other other approaches to modulate the microbiome could be the way ahead if we find the right tool. And that's why I shared with you this model where we can use it in vitro to screen big number of banks of potential prebiotics or probiotics or other modulators to identify ones with potential that we can carry and try to test them in vivo, for example. So yeah, that's another way to attempt. But I'm not sure if we can use this at an advanced, as Dr. Divine mentioned, we cannot, when a person has already peritonitis, at that stage we have to intervene surgically and in the clinical environment. But maybe to maintain a balanced microbial community from there, using a prebiotic, or if a person already has a normal biotic or balanced microbial community, we can maintain that by regular use of prebiotics, for example.
Yeah, I think that I sort of brought myself to a halt there, because I think one of the difficulties with periodontal disease is partly related to people's practices as well as their predisposition to the disease. So oral hygiene is really, really important. But there are also people who are more likely to get it. So you then maybe need to be looking at maintaining them. But I think that that there is we're looking at possibly people who are at risk of RA have a different healthy microbiome. We need to look at more people. We need to look to be certain that those sites aren't being influenced by the fact that there's disease in the mouth as well. But what we'd like to know is, can we actually correct that dysbiosis? Can we correct that to a more normal microbiome? And how can we do that? And how can we do it in a way that's maintained?
Right.
And that's quite an interesting challenge, and it's a really good question for the multiplicity of people who came up with it.
Excellent. Thank you both. Let's see if we can get in a few more questions. One question was directed at Professor Al-Habshi directly. How does the in vitro model dysbiosis index compare against clinical oral health assessments like MGI or pocket depth?
Yeah, I mean, that dysbiosis index that we calculated is only valid for the model because it was calculated based on the relative abundances in our clinical inicula. So it's not a generic dysbiosis index, and currently we are working on a more generic dysbiosis index that can be used to assess clinical samples and we are trying to see if that can be used as a marker to discriminate between peritonitis and healthy individuals, and we are still working on this.
Excellent. So this is work in progress. Another question is the question on alpha diversity and the association of an increase in biodiversity with health or disease in the oral microbiome. Now, just to elaborate a bit, for example, for other body sites such as skin, for example, we typically associate an increased alpha diversity with health. However, obviously the skin and the oral cavity are very, very different body sites with different ramifications of microbial colonization on the host. So would you like to comment on that? Do we expect an increase in alpha diversity in health as we find it with certain
peritonitis maybe is an exception in this context. All studies show that peritonitis is associated with an increase in diversity, which is in contrast with many other diseases in the context of the gut microbiome, for example, where they show that health is associated with increased diversity. So it's the other way around in the context of peritonitis.
But you can find studies that have found the opposite, or a change in, or a static, sort of similar levels of diversity. So not quite sure why, but again, if you search through the literature, you'll get more studies that show that the diversity goes up. But there are some that it either goes down or stays the same. But there is still a dysbiotic community. It's still different, and I think probably need to probe into what the differences are in those studies, in the way that they've sampled. I don't know why that is. And
then finally, thank you very much. I think I'll take one more question, and then we've used up a good fraction of time today. This is a question I think that is actually addressed at the Cosmos ID Analytics. And it asks, what fraction of identified microbial lineages do you classify at the strain level in a typical experiment? Yeah, I'll probably take that one myself. So thanks to this phylogenetic organization of biomarkers, the Cosmos ID platform is able to discriminate down to strain level. And we have strain level biomarkers essentially down to subspecies, and strain level for all microbial species that you typically find in the oral cavity. So essentially all microbes that for which high quality genomes exist in public databases, or we were able to aggregate high quality genomes through collaborations with biobanks, de novo sequencing projects, and so on. So in other words, we were very sensitive in detecting to subspecies and strain level for all the predominant species that you'd expect. However, the phylogenetic structure I was referring to not only identifies biomarkers that are unique, for example, at the lowest level of this phylogenetic tree of microbial life, so it's subspecies or strain level, but also biomarkers that are shared within the same lineages upwards the tree structure of the database. And that allows us, in cases where, for example, a certain strain has not been genome sequenced, is therefore not accessible or available in any database, to make nearest neighbor placements and identify the organism, for example, at species level. We also have recently expanded the Cosmos ID databases using what we call the dark matter database. Dark matter databases do not rely on high quality genome assemblies, but rather on hypothetical genomes established through metagenomic assemblies, so purely assembly derived genomes from microbial community DNA, essentially. And that allows researchers in cases where the strains cannot be identified and the nearest neighbor placement has been made, to do a secondary query against the dark matter databases to see if any genomes from organisms that couldn't be cultivated but that are, could only be assembled can be made. So in summary, there is very broad coverage of microbes, and certainly all lineages that you'd expect are present. However, we go beyond that by including also microbial genomes that come from metagenomically assembled genome projects. Any questions that Nazar or Deirdre you have spotted in the section that you'd like to comment on? If not, I would really like again to take a moment to thank both of you for that wonderful presentation, for a great ongoing collaboration. And I'd like to thank all the attendees today for joining us for this webinar, and we're looking forward to seeing you again in the next installment of the Cosmos ID webinar series. With this, a wonderful day to everyone, and goodbye.